Do you think?
So hello everyone, myself Dinesh Kumar. So now we will see how we can analyze this data set in different tools. The data set that we generate in sequencing, metagenome or AMR sequencing, gene sequencing, to the NGS data set. How we can analyze and visualize the data set so that it can be represented with the significance, whether it is abundant or it is not abundant, and how much it is present in different sample types based on our metadata. We have we have seen that from different bacteria species we have identified some AMR genes which are present in those bacteria, like in previous session. In Campylobacter, there were some resistant genes were there. So in broadly, data set can be of different types, whether we are identifying those genes from whole genome bacterial species or from environmental samples. So there can be different types of data, but for our project, we are looking from the Campylobacter and those E. coli and from poultry related samples. So just to brief about. So for a metagenomic study, what is our hypothesis? Before we go for sequencing, we should have some kind of hypothesis for experiment design. So the things start from there itself. Otherwise, our correlations or statistical analysis would not be appropriate if our hypotheses are not relevant. Sometimes, so the first point come here is the formulation of aims and objectives. So these you must have already covered all those aspects. So I will not go into the details of those things. We have already designed and we have already set objectives in our projects and studies. So, but these things come at the end also. If initially there is some issues and that has not been resolved properly in the experiment design, these things will come and reflect in the statistical analysis during the final results of our study. So that thing should be taken care of. Even before sequencing, there was a lot of discussion from whether we should go from for Illumina-based analysis panels or ion torrent-based panels. So that has been discussed thoroughly, and some experiments have also been performed and compared the data. In the morning session, also, Madam was briefing about Illumina, and one of the participants asked about which is better platform. So those things should be there before, because at the end of the day we have already invested our time, energy, and there has a lot of cost has been invested in those experiments. So after that we see that these things are not properly designed. That should not happen. So it requires sample collection. Here it has mentioned as a microbial samples, or then there is a sequencing, whether we go for a shortcut sequencing or whether we go for a targeted sequencing, whether we go for amplicon sequencing, that is targeted sequencing. And then taxonomic profiles has been generated from different databases against blasting against like in the previous session we have seen that against the Eastern AMR database that was done. So now we have to analyze the results. So broadly, there is a sample DNA sequence. But now, what is the significance of statics in these studies? So what is basically it is when we look at the data from different perspective, everyone has a different view. not all can understand what is the genome sequencing data if you ask for a layman person. But visually it can be better understood by a layman person also. Even in a bar graph, a very simple diagram, bar graph, that data can be understood easily that in which sample what is there. That kind of things, that is the use of statics here in simple terms. But when we add those parameters additionally with this statistical analysis and visualization, so then we can say, yeah, this sample it is abundant or it is better statistically significant compared to you. All of you must have heard about the p values. What is minimum p value? So the cutoff value that there you can call it as a significant. So zero point zero 0.05 normally in all expression data you utilize these kind of simple terms. So as I described as shortcut sequencing, in shortcut sequencing, it allows to capture the entire pool of genome from a sample. There, the functional and taxonomic information both are captured at the same time. While in the targeted sequencing like 16S sequencing, there is no functional aspect is being captured. But in shotgun, you can capture those aspects. But in advanced sequencing methods like metatranscriptomics and those, the functional genes can be sequenced. So to understand and visualize those significant features from our data set or our studies, we use a very simple graphical user interface tool, that is a STAMP. STAMP is a statistical analysis of taxonomic and functional profiles. So it is not only for taxonomic, you can features, you can of those functional features like gene numbers, gene abundance. Those features can also be represented in this STAMP. So STAMP was it is a window-based platform. In Windows, you can install this platform to analyze and visualize this your data set. And this was very it was developed and released in 2015. The last version came. So 14, 15 there is a publication from this. It also can be on from Linux based platform also. It can be installed and visualized. Now, what the STAMP requires? STAMP requires a profile from the analysis. So that profile can be obtained from MGRAS, and in previous analysis also you have done analysis for one sample. But if you have for 50 samples, so you have to create that profile for 50 samples in tabular form, in simple Excel or TSV file format. It can be used in STAMP. And along with the metadata. Now, metadata can be of different types. Like for clinical samples, you can add information about the gender, age, or disease, cancer, and control samples. So those kind of information you can additionally add with your sequencing information. So that information, like in control sample, you want to compare the control with the disease or control with the experimental sample. So that can be added with your sequencing data. Now, all most of these like MGRAST is also a online platform. So what kind of comparisons is supported by or statistical test can be done using the STAMP? STAMP, now you have different groups are there for multiple groups, like different cities are there. You have more than 2 cities, like in Gujarat from top 5 cities. So that cities, one example is there, like city. So if you have more than one metadata information, feature information, so that multiple group comparison can be there, can be done. Two groups and for two groups like disease and recovered or disease and demise patient or control groups. So different two group tests can be compared. So these are the different tests: ANOVA, Kruskal-Wallis test, and for two sample also you can compare chi-square test, Fisher extract test to compare between two samples. So you have the metadata information along with your sequencing data, and you have the different kind of analysis based on your objective. One is like you want to compare between two groups or two group based on like one is male and female also. Other group is you can different groups as per your objective. You can divide your data as per your objectives. We will see in some of the examples how we are comparing this analysis. So for two sample comparisons, there are different parameters that are being used, and there is a description of these parameters is being mentioned here, like I-square test. If there is a large sample experiment, large sample data set is there, so which statistical hypothesis or test is suitable for which kind of data, that is more important to understand. Why should we go for a to which statistical test, we do not know, but we have to understand, like G test is there. One example, now different examples are there, permutation. So these are different tests that you can select based on the different groups that you are comparing or analyzing from your data set. So here you can see there is a one bold is there. So this test is suitable for analyzing two group comparison tests when you have two groups, and what confidence interval you are selecting. Bootstrap is there. Now multiple test comparison, story FTR correction is there. So along with this statistical test, some corrections are also added to your data set to in the visualization. Now for multiple group test, what is being utilized? ANOVA test is there. Now other different, along with ANOVA test, you can multiple correction, FDR correction, story FDR correction. So what is being as per literature, what is suitable, that is highlighted in the bold. So now you have a different hypothesis you want to try and test. So what are the different ways in which you want to represent your data set? In literature also you have seen heat map plots are there. In simple taxonomic literature you can see heat map plots is there, scatter plot is there, bar plot is there, box plot is there. So now usually in bar plots are not accepted in recent literature because uh, they want the correction along with the simple bar graphs. So in high impact papers, no bar graph you will find generally if it is not represented along with the statistical errors. So those simple bar graphs are outdated now. So you have to keep updated with what is current literature say about what kind of graphical view that is perfectly suited for your data set. Scattered plots. So apart from STEM, there is another tool which is past. Past in past also you can calculate different PCO plot, diversity indices, beta diversity, alpha diversity. So these two tools are window based, and on local server you can install and do your analysis. Now there are different command line based tools are also there. As we know that in literature there are different beautiful graphs are being with high quality images are required for your manuscripts. So at that time you require a basic knowledge of R also. So because there are different R based programs and tools are available to visualize your data set, that are command line based tools. So here is I'm just showing you some of the representative packages of R which are utilized to visualize your data set with the statistical significance and tests that are represented. And in now some certain tools and these tests also have limitations also. Which package is suitable for what? Now it is having different. Now you can see minus sign is there. This means it cannot be performed in this package. and now here also you can see there is plus sign in all three. So this particular package can be utilized and compare and perform these different tests that are two group, multiple groups, and discrete factors. Similarly, DSEQ2, SR, and different tools. Now, where we can see in transcriptome dataset, you want to compare the expression dataset. So for that also, different tools are available. Now, and this is just I have explained. Online tools also. Now, every day now, lot of tools are being. You can find it out on the GitHub repository. In papers also, lot of everyday lot of tools are which are better and more user friendly. As most of us are not friendly to command line based analysis, these tools are being developed for a user friendly window based system is there. So that Shiny application is there. You can R packages, but Shiny application there are different transcriptome, meta-genome, RNA-seq. So all of those data sets can be visualized using these applications. But in the background, what is happening? They are using the R-based command, R-based packages or scripts are executed in the background in those command line base. Now microbiome analyst is there, Metagene assist is there, MetaVis is there. So these are all online web servers. There you can import your data set that is analyzed from the MGRAS profile or custom analysis that you have carried out the blast against the specific data set. Those can be imported into these online servers. So these are two key references. So we will have the hands-on session now. So in hands-on session, all of you go to that same login folder directory that was there in the data. So in that directory, there is these two different tools are there. Past is there and stamp is there. In the data folder,
in which recipe you can find those files.
recipe. So we have one example data set. So that example we will import into the STEM and we will see how we can create that profile.
वो सॉफ्टवेयर को आप अपने पीसी में ले लो, ट्रांसफर कर दो.
Drag it on the desktop. Past and STEM. Folder में पहुंच गए नहीं. STEM वाला folder है ना. Go to the STEM folder and there are two tools are there. One is STEM, another one is past.
दोनों की exe फाइल आप डेस्कटॉप पे कॉपी कर लो. एक्जीक्यूटेबल फाइल. मिला?
You need not write.
मिला सबको नहीं मिला फोल्डर फोल्डर मिल गया हाँ तो उसमें से है ना स्टैम्प और पास्ट दोनों अपने सिस्टम पे ड्रैग कर लो डेस्कटॉप पे You have to install the stamp first. And if it is not installed on the system, it will take only 2 minutes only, not even 2 minutes. Install the stamp in your system. Got the stamp? So if you have installed the stamp, open that stamp. Stamp open करो. Then if you have installed the stamp, open it. So we will import our data set, example data set in this. So from that folder only, what are other two files? I think there is no example data set in that. So we will import these two different files. So before that, we will see what is there in our file. We have two TSV files. In TSV file, one is a metadata file. So in metadata file, we will explore and see what is there in that metadata file. So this data set contained in the same folder, you will find these two files. So just before you just look at here, you can see. So in the metadata, we have 4 different columns are there. One is the sample. The first ID is a sample. So how many samples are there? There are 48 samples are there in this set. And what is that group? One is control, alive, and demise, and vaccinated and demise. So these are different groups are there.
You can refresh the IM folder. You will get 2 files over there.
आयन वाला फोल्डर पे
Refresh
चले
that
जाओ, उसको
folder.
रिफ्रेश करो, 2 वहाँ पे फाइल है, उनको आप डाउनलोड करो पीसी.
Refresh it.
So in that way,
दोनों को डेस्कटॉप पे ले.
आपको ये फाइल ओपन नहीं करनी है. Just you have to drag and drop on your system. Okay. So these two files we will import in the step. And how this file has been generated? The first file, the metadata file, you can see here. The metadata file, it contains the 5 parameters. One is the sample ID. Second is the group. I have made the group into different control, alive, and demise patient, and something like that. You can make these groups based on your experiment. Like someone is treated experiment is there, whatever that parameter that you have. Another parameter I have taken is the hospital. So the sample came from that particular hospital. So that one parameter I have added. So number of parameters you can add in your data set. But based on the objective, whether you want to analyze your data set from different cities or from different factors. Another is the location. All are from the Gujarat only. So from different cities, this parameter has been taken up. Otherwise, you can also add age, gender, but I do not know whether those objectives fit into your data set or not. But you can add multiple factors here. So those factors can be added and included in the analysis. Now, for each sample ID, there is a profile. So I will not save it. This one is the MGRAS profile that has been generated from the MGRAS. This is the taxonomic data set, amplicon sequencing data set 16S profile. So that this has been generated from MGRAS. So in this you can see this the domain level, phylum, class, order, family, genus, and species. So in the species, this is OTE table that has been exported from the MGRAS. MGRAS is online tool also. I hope all of you are aware and familiar with the MGRAS. It is not difficult. So from that profile, exported profile, now we will import this data set and we will visualize this in the stem. So this has already been done, but we will again see from how we can import this. Now you can try from your system. So go to the file. From the file, there is option of profile file. So in the profile file, you can select the
open your tab. Then
so from these two files, the first file one is the mgras covid.tsv file is there. So that file you can import. And in the second column, you can add the metadata.
tab profile. Sir, वापस बताओ tab profile. How to load data?
SEM profile में load data है ना, first column. Go to the first column from the load data.
So we have two options, profile data and metadata.
हाँ. In the profile you add the taxonomy profile. So these two files are just you have downloaded. That MGRAS file. That file you can add. In the profile. Group metadata, second metadata file is shared. That file you can import. So is there any problem? हाँ बट import तो हो गया फाइल, उसमें डिस्प्ले नहीं आएगा अभी, फाइल import हो गया ना.
हो गया सबका?
So from the parent level you have to select different. So first you go to the two samples. So there are you can see here. So you can see here like there are multiple groups is there, two group is there and two sample is there. So go to the two sample at this point. And from the two sample you select sample 1 and sample 2. There is sample 1 and sample 2. So in the drop down menu you can see different sample IDs that are imported in the data set. Can you see? All of you can see your data set or not? See, you can look at the screen also if you are not able to see your data set in your system. So there you can see on the right side top corner these three different metadata were imported. One is the group. Group, what was the group? Control, demise, and recovered. That was the group column was there. Second was the hospital. So from the different hospitals, those samples were taken, and you can see the profile from these different hospitals. So these three different colors represent three different hospitals that these samples are from those. Now there you can also select the location. So in location, this is a heat map plot, and this plot is at the order level. So you can also select that different level. Like if you want to see this at the family level also, you can see this abundance of in the heat map form, and like you can see this Pseudomonaceae family is abundant in those samples. In these samples, it is abundant, and these samples are from which location? You can see it from here. This orange color represents is Gandhinagar. This blue is Ahmedabad. So likewise, whatever the metadata that you have imported, you can see and visualize your data set. And now this is different groups was there, family level or species level you want to see. And this is in the heat map form, heat map plot. Scattered plot, you want to see the scattered plot, it can be generated. PCA plot also, it can be generated. So what does this explain? PCA plot, this variance in different samples can be explained with this form. Bar plot is there. You can select a species in the bar plot. So there are different species abundance is there. So this particular sample contains the abundance in the higher abundance. Now if you double click this species, so it is more present in this, and this sample is from Gandhinagar. So likewise that it can be represented. So extended error bar plot is there. So there also we have selected at the species level. At higher level of abundance, like now I have selected at the phylum level. So at the phylum level, these unclassified bacterial sequences are much more in abundance, and so based on our comparison group, what is that comparison group? Here is the location. So in which location, which sequences are more abundant? Here you can see it in Gandhinagar. Now you can configure these plots also. I think it is not showing lesion position is upper left, so there you can configure this plot where you want to see these lesions. So now it is there. Sometimes it is overlapping on the main figure, so you have to avoid those kind of things. So you can rearrange these lesions by configuring this plot. You can adjust the weight, and you can adjust the size of figure, marker size, or what is size of the figure you want to see. So error bar plot. So those things can be configured in the you want your figure that you final figure that you want to export. Now see here, you can see this lesion is overlapping on this main proportion of. So you have to avoid those kind of figures. You have to place it in such a way that it should not overlap with this figure, or sometimes some digits are missing and these are overlapped with this legend. Now how you want to highlight which bacteroids, or what you want to highlight in your figure, actinobacteria. you can select it from this. So this is in the different color, red color it is there. So the significant features you can highlight with this configured this particular species you want to highlight, or it is not represented here. Now you have to, let's we try to select in different in other parameter that we have used, like one group is there. So this group, from this group you want to see the scattered plot, or you want to see the bar plot. In the bar plot you want to see this species. So we are looking at the phylum level of abundance and sample wise. So there you can see in these samples from, they are control samples, and in control samples this particular phyla is highly it is much more abundant compared to the this group. So there is a clear cut difference in the this alive, control and demise, or for this particular species phyla we are looking at now, because we have selected, like you want to see the difference of firmicutes. So firmicutes are, they are not abundant here in this group. So these are control groups. So likewise you can import your AMR data. In AMR data also you can see such kind of differences. In the AMR genes, different genes, you can in place of these species or bacteria, you can have your AMR gene or the bacterial species, in which species it is abundant. So those kind of metadata can be manually configured and imported in this tool. So but what is the significance of this tool is that you can statistically compare those genes with the your metadata and your bacterial species. So that thing can be done here. So now if you want to see only for two samples, so for these two sample, this sample and these are sample IDs here, and in these two samples, so it is at the minor level of only just two samples you have, and you want to compare the difference between two these samples. So I have compared here like this. So that may not be there from these two cities. Now you want to select some features, additional features also. So you can add these features. You want to see the comparison between these three species only. So from these three species, these firmicutes or actinor proteobacteria. So likewise you can visualize and represent. Now where we can add different tests, statistical tests are there. Chi-square test, so that can be added. So what is confidence? 95% confidence interval, CI value. So that is confidence interval. So you can also filter the features from this. Sometimes you can filter the specific features or features based on the p-value. If you want to filter based on 0.05, or you want to increase the p-value, or like additionally 0. You can increase or decrease, and you can additionally filter the features from your data set. So what is that advantage is that it is more user friendly. You do not need to know the commands or R or something like, so that is very easy to use. Like just window, you can utilize this tool and use for the statistical comparison of the data set that you have based on the different metadata. Now, apart from this, we also would like to use the past. So, what is the purpose of the past? Past is also very simple. Excel, like it is very simple in terms of utility. These two tools are very easy to use. And on, you do not need to know the Linux कि मुझे Linux चाहिए, तो ऐसा नहीं है कुछ. इसमें विंडो पे है आपके पास तो भी ठीक है, और इसमें कोई हार्डवेयर नहीं चाहिए कि मुझे सुपर कंप्यूटर चाहिए, मुझे ऐसा कुछ इंस्ट्रूमेंट चाहिए कि जिसके वजह से मैं कर नहीं सकता हूँ, so that limitation is not there in these two tools. आप इसमें क्या कर सकते हैं कि यहाँ से आप you can add the metadata information, you can select the row and column. इसमें so from so the first column is the species, so name of the species is there, so from that you can just copy and row attribute. So go into the row attribute and paste your. Now we are also copying the sample, which sample from this species is from which sample, so that headers are also we are copying. So as I said, it is very easy to use. It does not require lot of hardware utility or anything. So now from this file we will select that abundance profile, that abundance taxonomic profile. It is computed based on that Silva database from the MGRAST. So Silva database is so now you have the sample ID and there is a species ID on this column and the abundance of this species in that sample. So how you are going to calculate the different diversity indices? So you can have diversity indices and you can simply see just by clicking on here you can see the Simpson index, Chow index. So different beta diversity indices are available for your data set. Usually in literature also you see these comparisons are done. So now you can see and compare and different note the differences, which is low in number and which is high in number, where is beta diversity is low or high, where what is the Simpson index of this sample. So this comparison is done. So you can see and you can use this table for writing up or how many texas are there in that sample. In that kind of calculations are done here in past and very easy to use. Now this past is also not only for this diversity indexes, it can also generate different plots, PCA plot, principal coordinate plots is NMDS plot is there. So different discrimination analysis. So based on your objective, you can select this statistical component or comparison, which comparison I need to select, clustering that can also be done. So different one way, two way ANOVA. So very easy and user friendly method it is there. Multivariate analysis, univariate analysis, correlation analysis. So those tools you select, you can select and plot different uh, using past. So past is also very user friendly. Now you want to see the in the form of these plots. So here also you can configure your images and export this in PDF or PNG or whatever suitable format you require for your that. These are simple things like you can see here a sample ID. If you can rotate or flip or as per your requirements, that can be done. Or if you want to plot here the dominance or Simpson index, so there also you can compare and see. So visually you can see which samples is having this particular index is higher. In that table you cannot visually see. You have to compare the values. But in this graph you can visually see that this particular sample is having this index is in higher. And in the graph setting you can just you can increase the thickness of the lines or numbers or this axis. You can, if you want to increase the size of this axis, or you can export. Now you can export this in the form of PDF. So you can export and save as PDF, whatever name you want to give it. So I think and they are of good quality images. Do not have to worry about that. Now we will see another tool, that is a online tool. So earlier also there were different tools. Metagene Assist was there, but now this another microbiome analyst is there. So here also you can import, and now let's go to the home page of this. You have the abundance table, that is in OTU table is there. If you shortcut profile, now you want to analyze compare the functional groups KO, KEGG, so COG, taxa enrichment you want to do, so that can also be done. Now we will see this profile. So there are some example files. So what is in example file? What is required in example file? Sometimes for some of the samples you have to prepare the metadata as required by this tool, because this tool have a specific kind of metadata that is required for them. Sometimes it may not be suited for that. So here you can select different taxonomic identity that you have utilized. Like if you have used a green gene database, so the taxonomic IDs can be given. If you have done with the Silva, that can be done. If the metadata of this file, it is there in the this taxonomy label, or you have to check this. And if it is like there are some errors are there, if it is not in the particular suitable format, so there is a difficulty and you have to reformat this. So there you can go and see a taxonomic profile. You can have some example profiles from here, like you have TSV file, Silva file. Now we will download one this profile. Similarly, we also required the metadata file. Sometimes metadata is included here. In this file, you can see the sample name or sample ID is there. So there is no requirement of metadata. But if you are providing the additional metadata file, so there is a requirement. we will see and import what analysis can be done using this example file. So this is a simple TSP file. From MGRAST also you can import this TSP file. For every tool there is a, like you can find in the, for every tool there is a particular format that it requires to import your data set. So in MGRAST you can import the biome file. So biome file, it incorporates the metadata. So different files can be imported in this. Now you have different, like you can see, plain text TSP file is there, biome file is there, Mothur output is there. So example data set is there. So from human from the green gene data set and database, there is one example is there. So in this example, so first thing it will compute that how many profiles are there, how many samples are there. So that computation is done. So data type is an abundance table. The format is biome. Annotation is done with the green gene ID, and these many numbers of OTUs are there. How many experimental factors? So 7 different metadata factors has been included here. How many counts are there? So it will just compute, and you can see the library size from the sequencing. Like these samples is having how many reads? Read count is there. So there is a difference of read in this. So that we need to normalize our data set so that can be statistical significance can be compared, because in this sample there is low read count is there. So what factor that you can utilize to normalize the data set? There is a higher count of reads is there. So you have to remove that biasness of read count. Sampling depth, that biasness can be removed by normalization. So So different factors are there. Low read count, minimum count of the feature. Here you can increase and decrease. There are 4 minimum count. These are different parameters that are given here. Prevalence percentage in samples, at least 20% of the samples should contain this minimum rate count in these samples. So that kind of normalization factors can be included here. So on other factor is low variance filter is there. So in this there are different standard deviation. Based on standard deviation you can differentiate. Interquartile range is there. So we are just using the different filters, default parameters. So there you can see there are these low abundance features has been removed from your data set. So to make them and to bring them on the same platforms, that is what is the meaning of the normalization. The minimum size of library, if it is 1 lakh read is there, so all should contain that minimum count. ऐसा नहीं है कि भाई एक में ज़्यादा है तो उसमें ज़्यादा abundance दिखेगा. So that is the biasness that is introduced. So you have to remove that kind of biasness. And in this, you can also see these are the R based R commands or scripts it is running. So sidewise you can see and save, and you can also execute in the R Studio. So that thing also, but we are not doing that now. Rarefication of the data set, data scaling on total sum scaling. So here you want to perform the rarefication. So normalization, this is the part of the data normalization. So you will submit this and it will filter out again the low variance features, low count features, and it will perform the data rarefication. So what we observed here is that now after data normalization and filtering, just like in command line from the raw data, you filter out the low quality data set, fast QC or print lite. That commands we can utilize and filter the low data count. Now we will see the rarefaction curve. So rarefaction curve also you can see. So based on the metadata like on the right palm, gut or tongue. So this data set contain these four features. It is represented here. Now, now filter count you want to observe. You want to so you can also see the bar plots. In the bar plot at the phylum level, there is difference of abundance in gut, this right palm or left palm and tongue. So they are comparing this microbiome profile of these four different gut, palm and tongue. So there is the comparison. Now, if you want to see at the class level, there also you can, that was there at the phylum level. So the differences at the class level also, and it is grouped into the factors. Like these are the different experimental groups that we have taken. So based on that, it is grouped. Or similarly, you can, if you want to merge, so the top 10 only or top 20. So there also you can select these features if you want to based on the total or median. So we will see based on the total, the top, there is some error, it is showing. So sometimes you have to select multiple combinations also. There again, now the same what input file we have given is the only taxonomic abundance. That is TSV file along with the metadata file that previously we have utilized in the past and stamp. Simple that file it has been imported or it can be utilized. Here we have taken the example file, but the same file can be imported after formatting because that is not formatted, therefore it was not imported. But the simple file now different rarefaction, phylogenetic tree, heat map tree, alpha diversity indexes. In that also we have seen, but it is better in terms of visualization. So that is the difference. Now there we were seeing the graph in that simple that count was there, some number was there, Java number was there, index number was there. But here it is represented in the graphical form. So definitely this is better compared to that. So these things are regularly updated. And there also you can download this in the PDF file format. And another thing is that what test it has giving, what is the p-value significance is there, and what another like you want to at which level of this it is computed, whether it is computed the genus level, species level, or phylum level. So that thing can be, and which diversity index you want to compare. Like there it is, Chow index is there. Simpson index it is there, along with which statistical method should be there. This test you want to apply. Colors you can select. And it is like you can see, it is filter count after normalization. It is not direct raw data. It is not representing in terms of raw data. That's filter count. So there also you can see. And what is the difference between these two figures? This is sample wise. There you can see the individual samples. And what is the here it is represented in the terms of groups. Now this samples has been grouped here in the cut. So this metadata, it is grouped based on the metadata information. So that differences can be, now here it is represented like this. Individual samples it is grouped, and in the form of PDF or SVG or PNG, or you can also download this results table. Now in what is there in the results table? Some values will be there, some numerical values will be there that are represented in the graphical form in this figure. So while writing explanation of this figure, you require certain values. Okay, what is difference between this and this, so how much difference is there, so that kind of things can be computed based on these tables that you export. Now that is alpha diversity index, heat map clustering. So again, this is another heat map that can be represented. So it is represented here at the feature level, or try to look at higher level, because if you go at the down level, these figures, this cannot be submitted for, like, you cannot read what is there. So that is one drawback of this online tools. But when you perform these tools on a command line or on a local server, those figures can be modified in terms of visualization. Like if you want to set the size of this, but here it is, there is no option कि मुझे इसका size कम करना है, तो यहाँ पे option नहीं दिया है ऐसा. So this is the limitation of online tools. Normally it is there, but in the other tool you can utilize there. This you can export. Sometimes you can export those tables, or that tables can be visualized in different tool. Have you heard about ClusterViz? ClusterViz also is there. So that there also you can ClusterViz. So we will try to look at higher level. See, not on the feature level, but we will see whether can we see some differences. So see, there you can clearly see and visualize that it is. And what is one advantage is there, that multiple metadata features has been plotted here. Subject, reported antibiotic use, number of days since this experiment has been done, 14 days, 17 days, month, year. So different, the number of, how many number of metadata features you have, that can be plotted simultaneously on the same figure you can see. But and apart from that, you can select different color schemes, and you can also use clustering different based on that different factors. So it will group on based on your factor selected year. Another important aspect is that you want to see and select the patterns from your data set. So in pattern search also you can You can select the feature of your interest, like pseudomonas. You are interested in pseudomonas and you want to compare the differences between the pseudomonas in different groups or different experimental samples. So that features can be utilized here. Now LabC analysis is there. So LabC that can also be performed into command line based tool also. It is a command line based tool, but it is integrated in this platform. So here you can select your parameters, like you want to perform it at the family level, and some descriptions are also given. You do not know how much log score I should give, so it is giving that there should be certain value, it should be cutoff value you want to set. So default cutoff value it is 2. So that way you can select these values. If you are not getting, sometimes you do not get at this particular score, you are not getting any significant features from your metadata. It happens a lot of time. So you have to change those parameters, like what minimum or maximum value I am getting this feature which is significant. So therefore it needs to be looked at. And apart from the graphical, now you also have a results table that is there giving you the numerical value of your results. So at what p value? Sometimes zero point 0.05 p value it is not like cutoff line. So what happened about 0.049? What you do with that? You are not counting it in your experiment because there is cutoff value 0.5. So if there is a some features which are at the significance value of 0.049, so you are ignoring those features because it is not representing your data set. Though it is there and there may be some significance, we do not know. But when we are applying the statistics, these features are neglected. So that is also a kind of biasness that is introduced by us. So that happens. So, but in this table you can see like 4546.63. So 0.63 will not come as significant because we are having a cutoff line of 0.05. So you have to look at in depth of your data set. Should we consider this as a significant or not? See 0.046 or 0.046, 0.068, it is a 0.08. So what do you do with this? Is it not significant? You have to look at those values and significance of whether this species may be on in your data set. What is the role? So those explorations need to be done if you really apply the statistics with them. Some now see pseudomonas, it is not there, but and let us see in this graph also. So we have selected year as a factor. So in 2008 and 2009, this abundance that is filtered count in row. So it is log transform value, it is there. So in 2009, this value has increased of this particular family in the data set. So as I said, based on your data, metadata, you can decide the factors like whether gender is significant for your experiment or not, or other different aspects. Age, that is there significance of that. Are you studying that factors? Location, are you considering the location as a factor, or it is due to the location? Is there a variation in the taxonomic abundance or functional profile, or different antibiotics? There may be some differences. So those differences can be taken into consideration when you look at these plots. I do not know it. This graph is not coming, but sometimes you can see this data in the graphical form also. So dendrogram analysis. See, all there is certain kind of statistics that is applied in all these visualization that is happening in the background. They are not directly represented. And if your experiments are like, you can see there is a 8 and 9, let us, I think we need to change some this year to some different factor. So there subject, let us see what is the subject. Subject 1 or 2, antibiotics. Yes or no? There is a, so in dendrogram clustering, this no or yes means this should cluster at the in the same dendrogram, like this particular region. They should have the same experimental factors. So that is the significance of this. If they are not clustering, here you can see there is a mix of red and green. So maybe there is some issue with these samples, or our factors may not be suitable for this calculation. That one factor what we have considered here is the age or sample type. Let us see the sample type. I think it will be better. So we have to look now. See, in this sample type, you can see there is some difference. Some difference means these red colors are clustered together. So what is that sample type? That gut. So there is a similarity. Now you can see this blue one also. So that is tongue. There is a similarity and they are clustered together. So that observations we have to look at. If that is not happening, now this tongue and this they intermix. So that means there is some issue with this data set. Now you can see left or right hand. So they may have the similar profile. So we have to correlate our data set and compare those metadata, and then only we can see whether those experiments that we have carried out are of some kind of significance. So these kind of studies or methods should be applied to significantly look at our data set. Now we will see. Apart from this, now you can also here you can also select a different distance measure. These indexes can be also selected. Level of taxonomy can also be selected. So those factors also can be considered. Now we will see other random forest or taxa for fun. Now these features have been recently introduced. Taxa for fun is this is functional analysis based on the 16S profile, abundance profile. So earlier you need to, but it is not. It is based on the predictive model. Correlation network analysis also we can. So we can select different algorithms, Pearson correlation coefficient. Now some you can also read. You have to read along with this which parameter is suitable for what kind of analysis. So which algorithm is suitable for our data set. So that kind of things we have to see. You can look at the p-value threshold, sample type or the phylum. So we are looking at this. Now all features so cutoff count we need to reduce, because in this particular data set there is a problem with this cutoff count, or when we are applying this particular algorithm it is not giving at the as the output. So we have to change this threshold parameters. So So I have just explained you the simple or easy to use methods. Lot of literatures you have to also read. So, but they do not require that high-end computational facility. It can be performed on the laptop also, and but they are of lot of importance or significance. To conclude, based on your hypothesis, on your design of experiment, or your outputs that you are getting, on what basis you can say that I am getting Pseudomonas is significant in our experiment. So that can be concluded based on this statistics. Otherwise, 0.05 is not significant for me also. Because 0.4049, if you are setting a cutoff of 0.05, you are ignoring 0.049. So we have to look at those aspects. So I conclude here. Promise you can.
Okay. थोड़े सारे हैं, 5-10 मिनट में आ रहे हैं। वो बताइए,
So we can ask
कौन सी टेस्ट अप्लाई कहाँ पे करना है?
वो नहीं पता।
ये जरा बहुत सारा ऑप्शन है, लेकिन आपके थोड़ा सा पता हाइपोथेसिस के अकॉर्डिंग आपको कौन सी ग्रुप चाहिए, कौन सी टेस्ट करना है?
मल्टीपल ग्रुप
में है ना, टाइम पे नहीं है, टाइम पे है ना वो ग्रुप कंपैरिजन बैच बी टेस्ट क्या चाहिए, मल्टीपल ग्रुप के लिए फार्मोनोवा है, एनोवा है। एनोवा तो कोई नहीं यूज करता, फार्मोनोवा यूज करता है सब लोग। थोड़ा ज्यादा वाले भी अब एनोवा तो नहीं मानते, फार्मोनोवा यूज करने को बोल रहे हैं। इसमें जो एफिक्यूस है इसके वहां पे है ना, एफिक्यूस में सारा लिखा है कि ये टेस्ट है तो उसका इंपॉर्टेंस क्या है और उसको आउटपुट है उसको रीड कैसे करना है, सब उसमें दिया हुआ है। लाइकवाइज स्टैम्प है तो स्टैम्प के वीडियो में भी सारा एक्सप्लेन किया हुआ है।
You can also refer these manuals also. I think I have shared in that folder. I do not know if you want. Sorry, material कैसे share करना है? तो कुछ manuals है या कुछ भी है तो
मैनुअल तो आप ऑनलाइन भी डाउनलोड कर सकते हो। जो स्टैम्प का यूजर मैनुअल है, पास टेबल सब तो ऑनलाइन भी अवेलेबल है, और स्टैम्प है पास टेबल तो फ्री अवेलेबल सॉफ्टवेयर, आप चाहे तो अभी भी डाउनलोड कर सकते हो। ये सर्वर है तो कहीं से भी एक्सेस।
ये ऑनलाइन है, ये तो कहीं से भी होगा, वो सब अवेलेबल।
सॉरी
server free access for us. It will be helpful for
server
access for us. And, like,
our server. No, it is not possible. You come here and analyze your data. That is possible. उसको बुला देना, ये बदरेश कहाँ पे है? नहीं, उसको ये साइड लेक का बताना है ना, साइड लेक का बताया?
अभी नहीं बताया।
आप यहाँ पे बता दो ना, सर आए ना तब तक तो GBRC का साइड लेक की जो फैसिलिटी है वहाँ पे बुकिंग आप कर सकते हो, कोई भी इंस्ट्रूमेंट यूजर।
So you can all of you can go to prayer lab and say.
डेडलाइन बिजनेस सर्विसेज में स्टूडेंट के लिए 50% डिस्काउंट है। तो मैसेज पे पूरा रन करना है, अब स्टूडेंट डिस्काउंट में आपको मिलेगा। सपोज 1.5 लाख का रन है तो आपको 25,000 भी सिंपल है, उसको माइक्रो
Ours is one of the very cost effective facility, especially for the students.
yes।
You won't get anywhere this high end sequencing at such a low price.
For student it is 50% discount. For government organization it is a cost to cost. For industry it is 1.5 times cost.
It is cheaper even for industry. If they outsource to the different industry, they will charge more than at least 3 times they charge at least.
so what is the just web interface tool for WPS data analysis. This is from meta-genomics.
This is statistical analysis of meta-genomics data only, not for the examination in your reads and like. So this is just for the statistical